Assalamu alaikum. So in this lecture, what I'll do is wrap up like everything that we've just talked about, especially the characteristics, chemical characteristics of DNA, gel electrophoresis, dot blotting, sudden blotting, and put them all in action and see how we can utilize all of these concepts in something known as RFLP, or abbreviated as RFLP, which means it's an abbreviation for restriction fragment length polymorphism, and I'll explain that. So, but before we go on, let me introduce these enzymes, these magical enzymes. They are known as restriction endonucleases. Now, a nuclease is an enzyme that degrades nucleic acids. Okay, so a nuclease is an enzyme that degrades nucleic acids. So what it does is that it cleaves nucleic acids, breaking them up into pieces, different fragments, and so on. Okay, so it's almost like if I say an enzyme like saccharidase. This is an enzyme that cleaves or breaks down sugars, saccharides. If I say lipases, these are proteins that degrade lipids. Proteases, enzymes that degrade proteins. Esterases, enzymes that remove ester bonds, and so on. Okay, so a nuclease is an enzyme that cleaves or breaks up or breaks down nucleic acids. Endo means inside. So basically you have an amino acid, okay, so you have an amino acid, so you have a nucleic acid like this, and the enzyme cleaves it in the middle, not exactly in the center, but within the nucleic acid. Okay, so that's what endo means. Now, the opposite of endo is exo, EXO, which means that it cleaves from either end. Okay, 5' or 3'. Restriction means that these enzymes, what they do initially, they were discovered because they restrict the growth of bacteriophages, which are bacterial viruses that infect bacterial cells. So they restrict the growth of these bacteriophages. Okay, so they're known as restriction endonucleases. So what these enzymes do is that they recognize a specific sequence of nucleotides within the DNA. And then, and this site, this region, the sequence of nucleotides is known as a restriction site. So it's a place whereby the restriction endonuclease recognizes. Okay, so for example, you have right here a sequence within a DNA fragment. It's C, 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 G, 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 and that's it. So the restriction endonuclease recognizes this sequence. Okay, so here's a restriction site again. So the enzyme cleaves within the restriction site, generating restriction fragments. Okay, now what the enzyme does exactly is that it breaks the phosphodiester bond that exists between nucleotides. Now, so there are different enzymes, restriction endonucleases, and notice that what they do is that they recognize palindromic sequences. What do we mean by that? Well, if you look at the restriction site for this enzyme right here, so this enzyme is EcoR1. It was identified in bacteria known as E. coli, and then the enzyme was given the symbols R1 to differentiate it from other restriction endonucleases that exist within the same bacteria itself. Okay, so E. coli bacteria have other different types of restriction endonucleases. One of them is known as ECO R1. E. coli, by the way, is a bacteria that exists in rotten meat, and it is the bacteria that causes fatality of people if meat is not, if meat is rotten. So notice what the sequence is. It's GAATTC. Okay. Now you don't have to memorize these sequences, by the way, and enzymes, but you will memorize this because we'll talk about it a lot. So that's ECO1. Now that's 5', 3', GAATTC. Now read the complementary strand from 5' to 3'. It's GAATTC as well. So you read it the same whether we're talking about whether we're reading the top strand or the bottom strand. And so these are known as palindromic sequences that can be read that are exactly the same on both complementary strands. Another one is called Hindu 3. Notice the sequence is AAGCTT. If you read the complementary strand, it's AAGCTT. Same thing. And the same is for the third restriction endonuclease, small 1, CCCGG. Again, the complementary strand CCCGG. So these are known as palindromic sequences. So restriction endonucleases in general, they recognize restriction sites that are composed of palindromic sequences. So what they do is that they recognize these sequences, restriction sites, and they cleave within these restriction sites. And they are very specific. Okay, so if there is a single change in the sequence of the restriction site, they cannot cleave the site anymore. So for example, you have this sequence right here. Okay, and notice it's GAATTC, again, GAATTC. Of course, now the enzyme does not say, oh, that's the top strand, that's the bottom strand. It doesn't matter, okay? So it's that's just restriction site as a whole. So you can flip the DNA and still GAATTC. So it cleaves the restriction site or the DNA within the restriction site. Now, this site is recognized by the restriction endonuclease, by ECO R1, and it's cleaved, generating two fragments, left and right. Now, look at variant 1. It's GCATTC. So there is a change in here. There is a mutation, there is a variation, whatever. Okay. So it's GCATTC. It's not palindromic anymore. That's one. 2, it's different. It's not GAA, it's GCA. And in this case, ECO1 cannot cleave this sequence right here. And the DNA stays intact. So that's really important. That's why these enzymes are very powerful. The thing is, so let's say that you have a person. Of course, every one of us is diploid. Our cells are diploid, meaning that we have DNA from our moms and dads, and they can differ. They can differ in the sequences of DNA. So our sequences are not, our DNA sequences are not identical. They are highly similar. They are like 99.99% similar. So if you have a person having, of course, being diploid, having chromosomes from mom and dad, and within these sequences, the DNA within the sequences within the DNA can differ by one nucleotide, by one base. So, for example, let's say that this person is, of course, diploid, having two DNA coming from mom and dad, So that's allele 1, allele 2. We already talked about that. And they are 99.99% similar in the DNA sequence. But there is one restriction site that is different right here. So here it exists in this chromosome, but it does not exist on the other chromosome. So that's an allele, okay? So they differ. So I say that a person is heterozygous for, and I have to say specifically for what? For this site right here, okay? So in this case, the DNA of this person, if I take the DNA from that person and I add the restriction in the nuclease, the restriction in the nuclease would make a cut right here, and right there for the first chromosome, and for the other chromosome, it would make a cut right here and right there, and that's it. So it would not be able to make a cut right there. So in this case, the DNA of that person, this chromosome of that person, would generate 4 DNA fragments. Okay, and for the other DNA, that is from the other chromosome, the restriction in the nucleus would generate 3 fragments. Okay, so in total, the person would generate 7 fragments. But notice that the length of these fragments may differ, or they might be exactly the same. So the length of this fragment right here is exactly the same as this one right there. Okay, because you have a cut in here. The length of this fragment right here is exactly the same as the length of this fragment, because you have the cut made in here. But the length of this fragment right here in the middle is totally different. It's quite long. Okay, whereas here you have 2 smaller fragments. Now that's a variation. Okay, this is what we call polymorphism. Morph means like shape, poly means multiple. So there are multiple shapes of the DNA fragments. Okay. So these are the differences among human beings, amongst us all. We again have the same, we have very highly similar DNA sequences in our chromosomes, but we have some differences, and these differences create polymorphisms. Okay, so again, a person may have same alleles or different alleles at any particular location on our chromosomes or DNA. So we can be homozygous or heterozygous for any particular location, and that would generate different fragments, restriction fragments, that is. Now, and that's what restriction fragment length polymorphism means. That is RFLP. So basically, it's polymorphism, different lengths or different shapes, okay, multiple shapes of what? Of the length of what? Of the fragments, the DNA fragments. What DNA fragments? The DNA fragments that are generated by restriction in the nucleuses. Okay, so let's say that I take DNA from 2 individuals and I add a restriction in the nucleus to this one right here, to the first person's DNA, and the same restriction in the nucleus is added to the DNA of the second person. The restriction in the nucleus would generate fragments of different lengths, because there are subtle differences between in the DNA sequences between the 2 individuals, the DNA of the 2 individuals. Okay, and this is what is known as molecular fingerprinting. That is, each one of us has his own or her own fingerprint. Okay, and it is specific for that person. Now, so the same exists for any person's DNA. We all have we all have our own molecular fingerprint. That is, If I add restriction endonuclease, my DNA fragments of certain lengths would be generated. If we add the same restriction endonuclease to another person's DNA, different fragments would be generated. Okay, so each one of us would have his own molecular fingerprint. Okay, in that sense. Now, except for whom, by the way? Identical twins, of course. They have the exact same DNA, so they would generate fragments of exactly the same length, by the way. There are exceptions, of course. Don't worry about it. Okay, so let's look at this example right here. So we have 3 individuals. Okay, 1, 2, 3. And for a particular location on the DNA of these individuals, there are 2 possible alleles. You have allele A and you have allele B, okay, depending on the sequence. So for allele A, let's look at this individual right here. This individual is homozygous for allele A. So this particular DNA fragment or location on the chromosome, the person is having the same exact allele on both chromosomes. So when a restriction site is added, when a restriction endonuclease is added, it recognizes 2 restriction sites on both chromosomes. Okay, so the restriction endonuclease would make 2 cuts here and 2 cuts on the other chromosome. Now, if we do gel electrophoresis, they will run according to, again, size, and this one here would run the slowest because it's the largest. Same thing for this one. These 2 have exactly the same length, so they run together as one band. Okay. Now it's not like it's not like the DNA fragments to say, oh no, you're from mom, I'm from dad. You know, we're going to be separate. It doesn't work this way. So the DNA fragments get mingled together, they get mixed, and they run within the same band mixed. Okay, so this one right here is the smallest, so it runs at this speed. Okay, now this one right here runs right there. Okay, great. So you have the generation of six fragments, but in the gel, they look as 3 bands. They run as 3 bands because again they have similar lengths, okay, between the 2. Let's say that a person is homozygous for allele B. Again, they look exactly the same, whereby the restriction endonuclease makes 3 cuts on each one of the DNA pieces, generating how many fragments? 8. Okay, but they would run together because they have similar sizes. So these 2 would run together. These 2 would run together. Same thing here. These 2 and these 2. So you will have 4 bands when we do gel electrophoresis. So this is the largest one right there. This one right here is right there. So it runs at the same speed as the one right there. And this one is the 3rd shortest or 3rd longest. Okay, so it runs right there. Okay, and the shortest DNA fragment is this one right there. Great. Now let's say that a person is heterozygous, meaning that this person has allele A on one chromosome, allele B on the other chromosome, allele B, sorry, on one chromosome, allele A on the other chromosome. So we would have the generation of 7 fragments, but they would appear as 4 bands because these 2 run together, these 2 run together because they have the same length, and these 3 run separately. Okay, so this one right here, so this one right here is this one in the middle. Okay, this one is a bit longer right there. This one is the longest right there. This one is the shortest right here. Okay, and this one in here is that one right there. Okay, so that's how it looks like if we do gel electrophoresis. Now, what if we do electrophoresis? What if we do southern blotting? That is, we separate the DNA fragments according to size, and then we transfer the DNA fragments from the gel to a membrane, that piece of paper that I told you about, and then we add a probe. Okay, what would we see in this case? Well, let's say that you have two alleles. So you have allele A and allele B. And we're using a probe that recognizes this sequence right there, specifically. Okay. And let's say that we make a cut by echo R1. So we have echo R1 cutting in here. 1, 2, 3. For allele A. For allele B, it makes two cuts, here and here. But it doesn't make a cut right there. So notice that if a person is homozygous for allele A, you would have only one fragment, or you would have only one band. And this band represents this fragment right here. Again, the fragment does not represent the size of the probe. It represents the size of the fragment that is recognized. Remember that. So it's almost like you have a light, you have a probe right here. Okay, it marks this individual, but the size of the individual is not represented by the size of the probe or the light on the helmet. Rather, that's the actual size of the individual, the whole thing. Okay, I hope it's clear. So let's say a person is homozygous for B. So this person is having both chromosomes having this DNA fragment. So the probe right here would recognize this whole long fragment. So it appears like this. Why? Because again, we have the DNA fragment run on a gel according to size. So this one's the slowest compared to this one right here. Okay, so it would appear on top of the gel or slash membrane. And when we add the probe would bind to the DNA fragment, whatever the DNA fragment is located. So it's located right here. So the probe would bind to this band. It runs to that point, so the probe recognizes this band right here. Now let's say a person is heterozygous, having one chromosome having this allele, the other chromosome having the other allele, allele B. Then the probe would recognize this fragment right here, and it's represented right there. And it would also recognize the long DNA fragment, which runs a bit slower originally, okay? So it's located on top of the gel slash membrane. I hope it's clear. That's if we do southern blotting. Okay. How can we use RFLP in the clinic? I'll give you a few examples. Again, we can use it to determine if a person is having a disease or not. We can also determine paternity, that is, who's the father, who's the mother. Okay, so it's used basically in forensic medicine. Okay, because each one of us, again, it has his own, her own molecular fingerprint. So we use it for diagnosis. So that's example number one. Detecting a disease known as sickle cell anemia. So sickle cell anemia is basically caused by a mutation in a single base. So instead of having GAG, rather the person would have GTG. Now that causes sickle cell anemia, which is something that a disease that we will talk about a bit in the biochemistry course. So in order for a person to have a disease, this person must have both alleles, that is both chromosomes, having GTG. A normal person would have GAG on both alleles, on both chromosomes. And a carrier would have one allele being GAG, normal, and the other allele having the mutation having GTG, a sequence of GTG. And it's symbolized as HBS, S for sickled hemoglobin. Okay, so hemoglobin, by the way, is the oxygen carrier in our blood. Okay, so these are the possibilities. You can have someone who's homozygous for normal alleles. That's a normal individual designated as A. A person being homozygous for the mutated allele, and this person is designated as an S for sickled. And a person can be a carrier or heterozygous, having one normal allele and one mutated allele. Okay, good. So we add a certain restriction endonuclease, whatever the name of that endonuclease is, and we can use a probe. And the probe recognizes this fragment right here, or this sequence right here. Now, if we add the restriction endonuclease, it makes a cut right here. 1, 2, 3. Now, the length of this whole thing is about 1.35 kilobases. That is 1350 base pairs. Okay, that is 1,350 base pairs. Now, if a person has a normal allele, then the probe would recognize, then the restriction endonuclease would make a cut right here, and the probe would recognize a fragment that has a length of 1.15 kilobases. If a person has a mutation right here, then the restriction endonuclease would not be able to cut right there, and the probe would recognize a fragment that has a length of 1.35. Okay? So again, these are the possibilities. A person can be normal, having both normal alleles. So both of them having a restriction site that is recognized and cut by the endonuclease. And if we do southern blotting, the probe would recognize the fragment that has a length of 1.15. Now, if a person is having mutation on both alleles, on both chromosomes, then the endonuclease would not be able to cut here. It would make a cut right there and right here, but not right here in the middle. And the probe would recognize the sequence that is 1.35 kilobase long. Okay, so it runs slower than 1.15. If a person is a carrier having both alleles, the one without a mutation and the other with the mutation, then the probe would recognize the long DNA fragment as well as the shorter DNA fragment, the 1.35 long kilobase fragment and the 1.15 kilobase DNA fragment. Okay, so this person is a carrier. Okay, so let's look at this paternity testing now. That's example number 2. Now, in this example, in terms of paternity testing, the DNA of children must come from both parents. Okay, so if we do RFLP, all DNA fragments, all restriction fragments that appear in the daughter's profile, genetic profile, and son's genetic profile must come from both parents collectively. So the idea here now is that we take mother's DNA, father's DNA, and children's DNA, and we add a restriction endonuclease, and the restriction endonuclease would make cuts everywhere, generating DNA fragments, okay, that are unique for each individual. That's molecular fingerprinting again. So if we look at the mothers now, you have the generation of 1, 2, 3, 4, 5 different fragments. If you look at the fathers, you also have 5 fragments. Now they have different lengths. Okay, I'm making it simple in here. So you have 5 fragments here, 5 fragments here, different lengths. So that's polymorphism. Well, let's look at this girl right here. The question is this girl the true daughter of this couple? Well, let's see. Now, all fragments that are generated from the daughter's DNA must come from both parents collectively. So this fragment right here comes from the mother's DNA, or it has the same size as the fragment that comes from the mother's DNA. Okay, so this fragment right here has the same size as the fragment that appears in the mother's genetic profile or molecular fingerprinting. So we can say that, it appears that this fragment comes from the mother's. Well, let's look at this one here. It exists also in the dad's profile. So we say, it appears that it comes from the dad's DNA. Well, let's look at this fragment. It has the same size as this one here, so we say it appears that it's coming from the mother's, and this one here has the same length as this fragment that appears also in father's DNA, or profile, so we say it appears to be coming from this man's DNA, and this one here also has the same length as a DNA fragment that appears in the mother's genetic profile, so we say, we can comfortably say that this is the daughter of these two individuals, okay, because all of her fragments also appear within the mother's and father's DNA profile or genetic profile or molecular fingerprinting. But let's look at this girl right here. She has this fragment. It has the same size as the fragment that appears in the mother's profile, so we say, looks like it comes from this woman. Well, let's look at these two. They appear to be coming from the father's DNA. Okay, notice that one of them is common. It's also present in the daughter's DNA, but the other one is not. So that's how it is. So because remember that 50% of DNA comes from both. 50% of DNA comes from mother, 50% comes from the father. But it does not mean that if a child has 6 fragments, that 3 of them would come from the father and 3 from the mother. So it's not like 50-50 for any particular fragment. Okay, let's look at these two. Now this one right here exists also in the man's profile. This appears in the woman's profile. So we say, we can comfortably say that this is the daughter of these two individuals. Great. Well, let's look at this boy right here. This fragment does not appear in the mother's or father's profile. It's different. It's a bit larger than this one here. These two appear to be coming from the father's DNA. And these two do not appear to be coming from either one of them. Okay, they're different. They have different lengths. So we can assume that this is the son of this man, but not of this woman. So probably he's the son of his first marriage. Okay. Well, let's look at this individual right here, this boy. The fragments are totally different than the profile of the fathers and the mothers. Okay, and that means that probably this boy is adopted. Okay, so this is paternity testing. Basically, children should have their genetic profile coming from both mother and father. Well, let's look at crime scene. I'm sure you've seen the show CSI, crime scene investigation, where basically, in almost in every show they say, we have his DNA. Once we have his DNA, we can tell who the murderer is, right? So that's how it's done, actually. Well, this is one way, RFLP. By the way, RFLP is an old technique. Probably it's not used anymore. But basically, you can it's used for a crime scene investigation. And the idea here is that if there is a blood stain, you can extract, you can isolate DNA from this stain, and you can do RFLP, again, fragmentation, separation by electrophoresis, you transfer to a membrane, and you add a probe that is specific to certain DNA fragments, and then you can see the profile. And the idea here, by the way, I'm sorry my picture is here, but you can look at the slides. But the idea here is that if we have a suspect and the suspect's DNA or blood is in the crime scene, then we should have a total match. That is, the unknown DNA profile should match the suspect's DNA profile. Okay, because of course. So let's look at these real cases. Okay. So here, that's DNA taken from the crime scene. Okay, and RFLP was done. This is DNA from the victim, because you don't know where the blood stain is coming from, right? It could be from the victim, it could be from the murderer, it could be, by the way, from the police officer when collecting blood. If he sneezes, for example, that's a crisis. This is a disaster, because now the stain is contaminated with another person's DNA. So police should be careful when collecting specimens from crime scenes. Okay, so this is we have two suspects right here. So we compare the profile, the genetic profile of these individuals with the stain or the DNA taken from the crime scene. So I think you can tell who the murderer is, or who the owner of the blood stain or the DNA taken from the crime scene is. So again, you can see that there is a fragment right here, another match, a third match, a fourth, fifth. So there's exact match between the DNA of suspect 1 with the DNA taken from the crime scene. So we can say that this is a true suspect. Now, if you look at this individual right here, the stains, although there are some similarities, but the stains are really different. Okay, so like this one, for example, right here, okay, it doesn't exist in the blood stain or the DNA taken from the crime scene. So you can say that probably suspect 2 is not the true murderer, it's probably suspect 1. Same thing here, we have DNA taken from the victim, and then you have the specimen right here, and then you have 3 suspects, right? 1, 2, 3, and you start making comparison. It's obvious that the DNA profile, the genetic profile, is different between the specimen versus victim. Okay, so totally different profile. Maybe there are some similarities, but there are differences. So you can comfortably say that no, they're different. This specimen does not belong to the victim. So look at the suspects right now. You look at, again, there appears that there is a match. These 2 also appear right here. This one here appears here. Same thing with these. So it seems that suspect 1 is probably the culprit, the murderer, or whatever. Okay, so that's how it is. Now, these cases are clean. Okay, usually specimens are not really that clean when collected, because you can have contamination. Let's say that there's a fight between the victim and the murderer, and there's blood that is mixed between the two. Blood of the 2 individuals mixed on the floor. So when DNA analysis is done, it wouldn't be that clean. It would be really messy. But you should, if there is a match between, there should be a 100% match between the specimen and the true murderer. Okay, so that's the point. The other thing is that you can have, let's say that you have blood that is that was lying on the floor for like a few hours, maybe 24 hours. You can have bacteria growing in the blood stain. So again, you would have bacterial DNA mixed with the murderer's DNA or the suspect's DNA. If the police officer has done a really lousy job collecting specimens, well, that's a problem as well, because in this case, DNA is not clean, and it would be difficult to interpret who the murderer is. Probably you have heard of the story of the famous American football player who probably he murdered his ex-wife and her boyfriend a long time ago, and he got away with it because his lawyers were able to convince the jury that the police officers were racist and they did not collect the specimens from the crime scene neatly, and they got it contaminated, and probably they mixed the football player's DNA with the DNA taken from the crime scene in order to associate the crime to the football player. So he got away with it, even though I think it was obvious that he probably killed his wife. Anyhow, a few years later, he did something else and he got into prison cell. Anyhow, so this is how forensic medicine is done, or even police work. That's how it's done. Allah